Variants of PCR Lecture 4. Colony PCR, multiplex PCR, nested PCR, reverse transcriptase PCR, quantitative PCR, hot start PCR, touchdown PCR, colony PCR, PCR screening of bacterial, E. coli, and yeast transformants containing cloned inserts. To determine insert size and/or orientation in the vector, the presence of an insert and its size can be determined by growing each colony in liquid. Plasmid purified by boiling or alkaline preparation protocol. Digestion of the plasmid with restriction enzymes that excises the insert. Separation by agarose gel electrophoresis. Multiplex PCR. Several pairs of primers annealing to different target sequences. This permits the simultaneous analysis of multiple targets in a single sample. Testing for genetic mutations, 6 or more amplifications might be combined. Analysis of microsatellites and SNPs. Nested PCR. 2 pairs of primers instead of 1 pair are used to amplify a fragment. In the first PCR, 1 pair of primers is used to generate DNA products, which may contain products amplified from non-target areas. Template, product of first PCR, in a second PCR, using 1 hemi-nesting, or 2 different primers whose binding sites are located, nested, within the first set, thus increasing specificity. Reverse transcriptase. Detect RNA expression levels. Qualitative study of gene expression. Detect gene expression through creation of complementary DNA, cDNA, transcripts from RNA. Quantification of RNA, by incorporating qPCR into the technique. qRT-PCR, RT-qPCR. Most powerful, sensitive, and the quantitative assay for the detection of RNA levels as compared to northern blot. Expression analysis of single or multiple genes. Expression patterns for identifying infections and diseases. May be either 1-step or 2-step RT-PCR. Real-time or quantitative PCR. Amplify and quantify targeted DNA molecule. Amplified DNA is detected as the reaction progresses in real time. 1. Non-specific fluorescent dyes that intercalate with any double-stranded DNA. 2. Sequence-specific DNA probes, oligonucleotides labeled with a fluorescent reporter which permits detection only after hybridization of the probe with its complementary DNA target. Applications. Rapidly detect nucleic acids that are diagnostic of infectious diseases, cancer and genetic abnormalities. Detect newly emerging diseases, such as new strains of flu. In the fields of food safety, food spoilage and fermentation, and for the microbial risk assessment of water quality. It is also used for the determination of zygosity of transgenic animals used in research. Hot start PCR. Hot start PCR avoids non-specific amplification of DNA by inactivating the Taq-polymerase at lower temperature. DNA is denatured by heating the sample at its denaturing temperature, and then the temperature is suddenly reduced to 55 degrees Celsius, at which primer and Taq-polymerase is added. Specific antibodies are used to block this Taq-polymerase at annealing temperature. When the temperature raises for amplification to 72 degrees, the specific antibody detaches from Taq-polymerase and the amplification with greater specificity starts. Touchdown PCR primers will avoid amplifying non-specific sequences. The annealing temperature during the polymerase chain reaction determines the specificity of primer annealing. The melting point of the primer sets the upper limit on annealing temperature. At temperatures just below this point, only very specific base pairing between the primer and the template will occur.